Welcome to this session of structure analysis. In this session, we are going to analyze the protein sequence primary structure, and we are going to predict secondary and tertiary structure. And we are also going to evaluate the predicted structure by using an evaluation solver. Protein structure, or say it can be explained in three levels, that is primary structure, secondary structure and tertiary structure. The primary structure is usually nothing but the amino acid sequence, order of sequence. So we can't predict primary structure, only we can analyze primary structure. Whereas the secondary structure means it talks about the alpha helix, beta pleated sheet and turns loops in a protein, which can be predicted from the sequence. Similarly, the tertiary structure, three-dimensional structure of the protein, can be predicted by using different solvers. After the prediction, the structure of the predicted protein may be evaluated by using different solvers like ProCheck, ERAT, and then ProWith tools. These type of tools are used to evaluate the predicted structure. Now we will go to primary structure analysis, then to secondary structure, then to tertiary and evaluation. Okay. So for primary structure analysis, we will choose for primary structure analysis. For primary structure analysis, we will choose for primary structure analysis. We will choose the tool known as proteinstructure. So it is available from proteinstructure. Is available from Xpazi actually. So you go to this website and then it will ask amino acid sequence of the protein. It should simply ask only the amino acid sequence. So now we will copy down the amino acid sequence of the amylase protein and then we will click the compute parameters and it will predict the primary structure. It will analyze the primary structures and it gives the information about the number of amino acids and molecular weight and theoretical PA value and then amino acid composition and the negative charge and positive charge molecules which is there, amino acid atomic composition. Like this, it will give excisions values and then stability status, how much time it will be stable, and whether it is stable greater than 30 hours. Like that, it is going to give their stability time, and then gravy value is also going to give. So this is our aliphatic index, and these are the informations we are getting it from the primary structure analysis tool ProParam. So this is how we can get many informations about primary structures by using these ProParam tools. Like this, we have several tools for analyzing the primary structure. For example, plot scale is another tool which is also used to analyze the primary structure. So once you predict the primary structure, you can save the file. So after carrying out this primary structure prediction, we can go for secondary structure prediction. For that also we have many tools for secondary structure prediction. For example, we have a tool of GAR and Chow-Forsman method is available to predict secondary structures, and then Coil's prediction server, JPRED server and SOAPMA server. We have several servers for predicting the secondary structure. Of this, we will go any one of the servers. Say for example, the Chow-Forsman server is available and GAR server is available, and then this APS2 server, it is available. So for prediction of the secondary structure of this server, we will also, I already told you that one more server is SOAPMA. SOAPMA server is also available for predicting the secondary structure. We'll explain with that. So now this is the SOAPMA server. So we'll go to the SOAPMA server and then here we can paste the sequence. When the sequence is pasted, we can submit the sequence, and after some time you will get the secondary structure predicted in this page. Then primary structure is analyzed, secondary structure is predicted, and now we will go to the prediction of tertiary structure. Okay, this is the result of secondary structure prediction. Here you can see that the sequence, submitted sequence, along with the secondary structure predictions are indicated here. So H represents helix, and the B represents beta bridge, and the E represents extended strand, and turns are indicated by green, and the orange color is again a bend. Chiral region is indicated by C. So this is how the secondary structures in a particular sequence are indicated. See, this part indicates that this sequence will form a helix-like. So this is how we predicted the secondary structure, and it is also give a graphical output of the structure prediction with different colors according to this helix, strand, and turns. With that, they are also using colors, and it's also represented in bar diagram format. So this is how we are predicted the secondary structure. The result clearly indicates that the protein will have more amount of helical structure other than the beta strand and other things. It will have a beta strand of about 28%, and then helix is around 21%. So almost the protein has highest value of extended strand compared to the helix region. So this is how we can predict secondary structure by using this SOAPMA server. So after we predict the secondary structure, the structure file can be saved. Okay, next we are going to predict a tertiary structure. The tertiary structure actually predicted in three ways. One is that homology modeling method, the second one is the fold recognition method, and third one is the ab initio method. So first one, we will go for homology modeling of tertiary structure prediction. In this, we can have several servers for prediction of this homology model. I choose CPH model for the prediction of homology modeling. So again, this is the page of homology modeling prediction server. Here you have to submit your sequence in FASTA format, and then after submitting your sequence, you have to click the button submit. So it will predict your tertiary structure, or we can give our mail ID so that we can get our output of this server in your mail ID. So before getting this result, we will go to another tertiary structure prediction tool, especially on the basis of fold recognition or threading. That is the second method which is used for prediction of tertiary structure, which is known as fold recognition server or method, which is used for predicting the tertiary structure. So Pyri is one of the famous server which is used for predicting the secondary structure, or we can use 3D PSSM, or we can use other servers like even Raptor X is also another server used for predicting the tertiary structure by using this fold recognition method. So for this method, it actually requires email ID for the prediction of the result. So you will give your email ID, and then we can give a heading of the protein here, amylase, and then you can paste the amino acid sequence alone. So you should remove the FASTA form region. Then you will submit only the sequence. Then it's a normal one. So you give a search. So it will give a results, and the results will be sent to your mail ID. So this is how we can predict tertiary structure by using this Pyri tool. And then the third method for prediction of tertiary structure is the ab initio method. And there are different servers are available for this ab initio method, and one of the famous method is ITASR, ab initio method. So one of the server is ITASR is a famous server which is used for tertiary structure prediction on the basis of this ab initio method. So in this server also we can predict. You can see the server. On this server also you can paste your sequence. it's better to use a faster format. So it's better we can use a protein which does not have a tertiary structure other than the protein, more common protein. Ab initio methods can be used for the protein for which there is no structure already predicted. So such sequence is selected for the study, that is known as cluster counting. And then this also will provide your results to the email ID. So it will ask your email ID, or you can simply submit the sequence. So they will ask a valid email ID for receiving the result. So you will give mail ID. Then so it asks you to register for the server submission. So if you register, it will give a password. So with that password you can submit your sequence. And then if you submit your sequence, again it will give error. So if you give a proper password, it will send the result to your email ID. So this is how you can submit your sequence to this iTask. So we submitted protein to this CPH model, and this model, the structure is predicted and it is available in the name of query.php. So pdb file, then you save this file. So now our structure of three-dimensional structure of the protein is predicted by this CPH model server, and the other two servers will provide results only to the mail ID. So with the mail ID we will get the structure file with the name of this pdb extension. It has different names, but it will end with this pdb. So that file to be saved as a 3D structure of a protein. So three methods are recommended. One is homology modeling, the other one is fold recognition, other one is ab initio. So when you should use this homology modeling? When your protein sequence matches with the structures which is available in PDB greater than 30%. That is, the already known structure, any one of the known structure of the protein matches 30% of your submitted sequence, then you can go for homology modeling. If your protein sequence matches only 10 to 30% of any protein which is available in PDB database, then you will go for fold recognition model. If you are not at all having any sequence which is matches your sequence not above 10%, only below 10%, then you will use only ab initio model. So this is how we can select any one of this model. Now we have a result with the homology modeling, and for fold recognition we get result through mail as well as with the iTask ab initio model. But all these three models are going to give a file with the extension of query.pdb. First we will see how it looks, the structure of the amylase predicted file. So for this we will use any molecular viewers. For example, we can use rasmol, rastop, chyme, and then a DS Viewer Lite, DS Viewer Pro. So what you are seeing now is a Discovery Studio viewer is actually DS Viewer, it may be called, utilized for Discovery Studio viewer is actually utilized to view the tertiary structure. There are so many tools are available for viewing the tertiary structure of the protein. So now we are going to see the structure of the protein with this tool, and then in the meantime we will check our protein structure whether it is correct or not by using a validation server. For that we will go for save server. So it is a structure validation or verification server. Okay. So in this server you can submit your file. So before submitting you have to choose your file. So we have already saved the file in our folder. So we took that file, and then we have to answer this question. After answering this question, you run all program file. So now you can see this, our job is started, and then we have to wait until our job is finished. After that you will get the output. Okay, so this is the different validation servers are used. You can see ProCheck, QuadCheck, ERAT, Verify3D, Prove, and then Redmall Viewer. All these servers are used to validate our PDB file. That is, around 6 programs are actually used for validation of our file. Here you can see this QuadCheck is actually finished our work, and ProCheck is running, Verify3D is running, ERAT is running. After completion of all these things, we will get our result in this page. So now I hope the structure is ready to view the 3D structure. We will see the structure now. So this is the predicted structure of amylase residue with this CPH model. It's a very neat and fine structure. So you can see this, the helices are indicated in red color, the strands are indicated by this cyan color, and then green color indicates the turns. Okay, so this is the important protein. You can see this very nice structure of the protein. You can see this core turns are surrounded by a helix. That is why it comes under the category of transporter nature. You can see a clear core. Since it has a helical structure, it can easily fix into the membrane. That's why the functional prediction server record it as a cation transporter. So that's how the structures will helps to study about the proteins and function. There is a structure-function relationship always exists. So these things can be analyzed, and their nature and an active site of the protein and the binding packets of the ligands are all the things are identified with this structures. Talking analysis, drug designing, everything can be used, everything can be achieved by using this structure. Thank you for this particular structure visualization. Next, we are waiting for the result of validation servers, and only we get the QuadCheck is reported. Other servers are not finished yet. So once these servers are finished, we will get the result in this page. It takes a little bit of time, I hope. Now it is, you can see when Verify3D is actually failed. It's not running. So after the result of all the server, we will get the result for the prediction. The result will shows that how much our protein is valuable. So to get immediate results, we can use some other server like QuadCheck server directly from the website of QuadCheck. See, this is the page where you check and prepare model. In this you can say protein model check. In this you can load our protein file. After loading the file, you click the send option. So now our file is running to this auto server. It will provide the result. Meanwhile, we will check this save server report. It's also going to it. It's not getting result. Now we will get the results of this QuadCheck server, and we can see the result of this what if server. So this is the result of protein structure analysis. In this, the server actually gives the information about the protein, and then they will say point-wise outputs. They will give errors where the residue, there is an error is present, and they will also give warnings about the protein structures. When you go down to the page, at the bottom of the page, you will have a block. Okay. So now you have two blocks. Structure of Z scores. When you get more positive values, the structure is better. Similarly, the RMZ score, when the score is very close to 1, it is better. So according to this RM score, our protein values are very much close to 1 according to these 2 points. But when you come to this part, we are having more negative charges. Okay, but the negative charges are less than 1 in many cases. So almost the structure is not too bad because it is not having any data like that. So in the predicted structure is found to be averageably acceptable one. So this is how we can validate the predicted structures by using this what if server. Through the same server, we can have the output of different servers in one place. By using this what if server, you will get only the result of what if server. So we explain both the cases. We can get information from other server, or you can get information from single server. So both the options are explained to you here. Thank you.